what if the genetic seeds for complex neuropsychiatric conditions, things like schizophrenia or autism, what if they aren't planted in the fully formed adult brain at all?
Right. What if they're planted much earlier?
Exactly. Like in the very first few days of a fetus's cellular development.
Yeah.
It just completely flips how we think about mental health in human biology.
It really does.
So today, we are taking you on a deep dive into this absolute monumental undertaking called the NeMO Analytics project. And our mission today is to look at how researchers have essentially mapped out the universal blueprint for building a mammalian brain.
And honestly, it is a massive paradigm shift because for decades, the central question of neuroscience has been, how do you start with a handful of completely generic cells and through sheer biological choreography, self-assemble the human neocortex?
Which is basically the control center, right?
Right, the center of our cognition, our emotions, consciousness. And to figure out exactly how it gets built, the researchers behind this project didn't just study one human brain.
Mm-hmm.
They aggregated single-cell data from roughly 200 different studies.
Wow, 200.
Yeah. We are talking about mapping individual cells across mice, macaques, humans, and even lab-grown mini-brains.
Which sounds amazing until you realize what a logistical nightmare that actually is. Because before this NeMO Analytics project came along, all of this cellular data was just scattered across the internet in unlinked public repositories.
Well, it was a mess.
Right. The metadata was messy. The formatting was totally inconsistent. It wasn't like putting together a jigsaw puzzle. It was more like trying to translate an ancient forgotten language, except every archaeologist in the world is using a completely different, incomplete dictionary.
That is a perfect way to describe it, actually. You have cells from a mouse and cells from a human, and on a molecular level, they just do not speak the same language.
Right.
So if we are going to move away from studying isolated single data sets and move toward this unified cross-species understanding of how brain cells are born and mature, well, we first had to invent a biological Rosetta Stone.
Okay, let's unpack this, because that Rosetta Stone comes in the form of two massive analytical tools. Because if I am tracking with you, the first problem is just figuring out what we are actually looking at, right?
Exactly.
So the researchers use something called metamarkers. And I like to think of this as sort of a universal name tagging system.
How about that analogy? Yes. A metamarker is essentially a composite expression of genes that shows up reliably across many different studies, completely regardless of the species.
Okay.
So, for example, there is a specific gene called EOMES.
EOMES.
Right. So if a cell in a petri dish is expressing a massive amount of that EOMES gene, the metamarker system basically slaps a universal name tag on it that says, hello, I am a neurogenic intermediate progenitor cell.
Got it. And it doesn't matter if that specific cell was swabbed from a macaque monkey or a house mouse.
Exactly. We now have a baseline vocabulary. It gives us a coarse consensus so we can, even begin to compare these vastly different biological data sets side by side.
But here's where I get a little hung up.
Right.
Once you have everything labeled, you have to actually combine it.
Right.
And if you use a mathematical algorithm to just mash all this mouse, macaque, and human data into one giant matrix, don't the unique human-specific details just get completely washed out by all the mouse data? Doesn't the human signal just turn to static?
Yes, and that is the exact trap of standard data integration pipelines. They force all this diverse, beautiful biological data into a single unified space, which absolutely blurs the nuances.
Okay.
But the researchers avoided that by using a different algorithm called structured joint decomposition, or SJD.
Okay, so how does SJD actually work to prevent that blurring?
Think of SJD like trying to find the underlying melody across radically different musical arrangements. Imagine you have a symphony orchestra, a jazz trio, and a heavy metal band.
Okay, quite the lineup.
Right, and they are all playing a cover of the exact same core song. If you took their 3 audio tracks and just blended them together into one file, you would just get a chaotic wall of noise.
Just totally unlistenable.
Exactly. But SJD doesn't do that. It listens to each track separately and extracts the shared dimensions of variation.
Ah, so it finds the hidden underlying melody, the core molecular mechanisms, without forcing the jazz trio to use the same instruments as the symphony.
Precisely. The data stays completely heterogeneous, but it becomes biologically linked. We can see the universal tune of a developing brain without losing the unique biological quirks of the individual species playing the song.
I really love that. Okay, so just to do a quick recap for you listening, we have our 200 messy data sets. We've slapped universal metamarker name tags on them so we know who is who. And we've used our SJD algorithm, our music producer, to find the shared melody.
Perfect summary.
So let's look at the actual blueprint. How do you build a brain from scratch?
Well, by looking through that SJD lens, the researchers found 7 shared transcriptomic patterns across all these mammals.
Okay.
But there is a very specific 4-stage assembly line for neurogenesis that is absolutely critical here. It starts at the very base of the developing brain with a pattern called P5.
The radial glia cell. These are the early ventricular progenitors, right? Like the absolute foundational stem cells of the cortex.
Yes. They are the factories. And once a cell is born there, the assembly line moves it to the next phase, pattern 4, or P4. This is the intermediate progenitor stage.
So P5 builds the base, but a stem cell can't just jump straight into being a fully functional neuron.
Right, no.
The P4 stage is the middleman. The cell has officially committed to becoming a neuron and is taking its first real transitional step.
Exactly. Which brings us to pattern 7, key 7, the nascent neurons.
The newborn baby neurons.
Yes, but there is a biological weirdness here that we really have to talk about. The data shows that the P7 stage is highly enriched in cell cycle genes.
Wait, like genes associated with the phases where a cell is actively copying its DNA to divide.
Yes.
But neurons don't divide. Once you have a neuron, it's just a neuron.
I know. It is a brilliant observation, and it puzzled researchers, too. But they used a highly sophisticated algorithm that acts essentially like a time-lapse camera.
Oh, cool.
Yeah, it traces a cell's developmental journey backward through time. And what that algorithm proved is that this P7 state is strictly transient.
Meaning it's just a phase.
A very brief, highly specific phase.
Right.
Even though these nascent neurons are expressing genes that make it look like they are preparing to divide, they are actively, rapidly shutting down their precursor state.
Wow.
They are closing the door on being a stem cell forever.
And finally, they graduate to pattern 2, or P2, the maturing neurons. This is the permanent adult neuronal signature that they're going to carry for the rest of their lives.
Right. And we have to be really clear about the mechanism here. This progression, P5 to P4 to T7 to P2, is not a series of abrupt switches.
It's not like flipping a light switch.
Exactly. A cell doesn't just wake up one morning and decide to be P7. It is a process of continuous overlap, often called neuronalization.
Sort of like a crossfade in audio editing.
That is exactly it. Even early on, those foundational P5 radial glia cells have a low baseline hum of neuronal genes. And over time, that hum slowly turns up in volume while the stem cell precursor genes slowly fade out.
And the physical timeline of this crossfade is just wild. The tracking data shows that it takes about 1 single day for a dividing cell to race from P5 through the middleman P4 stage and into that weird transient P7 nascent neuron stage.
Right, 1 day.
And then it takes another 3 days for the P7 pattern to fade out and the mature P2 pattern to fully take over. 4 days total to lay the foundational identity of a brain cell.
But the implications of that tight 4-day window are staggering, especially when we look at human health and disease.
Okay, here's where it gets really interesting, tying it back to what we talked about at the very beginning. The researchers took this exact assembly line and they mapped human genetic diseases onto it.
And the split they found is incredibly stark. They discovered that genes associated with gross physical brain structure diseases, conditions like microcephaly, where the brain is abnormally small, those are heavily concentrated in the early neural progenitor genes.
So the P5 and P4 stages.
Exactly.
Which logically tracks, if the physical foundation of the factory is cracked on day 1, the structural integrity of the entire brain is completely compromised.
Right.
But what about the later stages?
The genes most highly expressed in the later P7 and P2 neuronal stages are heavily linked to neuropsychiatric risks. This is where we see the genetic markers for schizophrenia, autism spectrum disorder, bipolar disorder.
Well, wait, here is the paradox. We just established that these neuropsychiatric genes are the defining traits of an adult, mature neuron. They peak at the end of the assembly line. But that's not when they first appear, is it?
No, it's not. And this is perhaps the biggest aha moment in the entire compendium. The genes that define adult neuronal function, the ones carrying the heavy risk factors for psychiatric disorders, first appear at very low levels in those early stem cell precursors. They are part of that early baseline hum we talked about.
So if a standard genetic test is only looking for where a gene is most active, meaning in the adult brain, It completely misses the origin story.
It completely misses it. Standard testing heavily underestimates how crucial the first few days of embryonic development are for mental health decades later.
That is wild.
The biological vulnerabilities for schizophrenia aren't just an adult problem. They're being quietly baked into the cellular identity during that initial 4-day neurogenic crossfade.
That is completely mind-blowing. Okay, so doing another quick recap for you. We've got the P5 to P4 to P7 to P2 pipeline. The psychiatric risks are baked in much earlier than we thought. But if mice, macaques, and humans all share this exact same P5 to P2 assembly line...
We're all playing the same 4-chord melody, yeah?
Right. So why is the human brain so vastly more complex?
Yeah.
Why are we sitting here debating genetic transcriptomes while a mouse is, navigating a cardboard maze for cheese?
Well, to figure out why our brains are bigger, the researchers needed a magnifying glass. They took their SJD algorithm and cranked up the resolution.
Okay.
They moved from a broad 7-pattern decomposition down to a hyper-detailed 40-pattern decomposition to find the molecular fine print.
Oh, wow.
And the absolute start of the show at this resolution is pattern 27.
Pattern 27, P27, the primate upgrade.
Exactly. When we look at primates, monkeys, and humans, P27 is highly expressed in a very specific, physically distinct type of cell called the outer radial glia, or ORG.
Okay, what is an outer radial glia actually doing?
Remember those P5 stem cell factories at the base of the brain?
The early ventricular progenitors, right?
Yes. Outer radial glia are essentially an entirely extra layer of stem cell factories that sit above them. They are highly proliferative, meaning they divide and conquer rapidly.
Well, I see.
They are the primary biological driving force behind the massive, dramatic physical expansion of the primate neocortex. They pump out the billions of extra neurons that give us our cognitive edge.
Okay, let me push back on this just a little. If outer radial glia and this P27 pattern are the secret sauce for primate brain expansion, what does the mouse DNA look like? Is there just a literal blank space in the mouse genome where P27 should be?
This is where we get into the brilliant efficiency of evolution. The answer is no. Rodents actually do have the P27 genetic program.
Wait, really? Mice have the primate upgrade?
They have the code for it, but here is the critical difference. In a mouse, the P27 molecular pattern is strictly limited to gliogenic precursors.
Meaning what exactly?
Meaning the mouse biology only allows that genetic program to make non-neuronal support cells. Things like astrocytes, the glue and maintenance crew of the brain.
Yeah.
The mouse does not use P27 to make actual neurons.
Oh, wow. So evolution didn't invent a completely brand new neurogenesis gene from scratch just to make the human brain bigger. It's just the ultimate recycler. It took an existing genetic program that the mouse was using for basic maintenance and repurposed it to pump out billions of new neurons in primates.
Exactly. If we connect this to the bigger picture, evolution frequently upgrades biological complexity not by drafting new blueprints, but by deploying old genetic programs in entirely new spatial and temporal contexts.
Okay, so thanks to this repurposed P27 program, we have this massive, expanded primate cortex full of billions of neurons. But the neocortex isn't just a giant uniform blob of cells. It has highly distinct functional layers. Kind of like a cake.
Yes, layers are crucial.
Once these newly minted neurons migrate from the stem cell factories out to the edge of the brain, how do they physically know which specific layer they are supposed to become?
Right. To solve the layer mystery, the researchers shifted their focus to adult human neocortex data. They ran another joint decomposition, another zoom level on the SJD algorithm, and they found 20 distinct neuronal maturity signatures that map perfectly to the different layers of the cortex.
And this is where the biological timeline we've been talking about completely shifts, doesn't it? Because earlier we said a cell goes from a stem cell to a nascent neuron in just 4 days.
Right.
But layer identity, that is a radically different ballgame.
It is a game of incredibly protracted maturation. Historically, scientists use simple transcription factor markers to identify cell types. A gene called RRB meant layer 4, or CUX2 meant the upper layers. But the problem is those simple markers peak incredibly early in fetal development.
They are just a rough draft. They tell you the general zip code, but not the actual identity.
Precisely. When you look at the full 20-pattern transcriptomic signatures of true functional layer identity, you see that they take years to fully mature.
Years.
Think about your own childhood and adolescence. The reason human children take decades to fully mature into functioning adults, unlike a mouse that is fully mature in a few weeks, is literally written into these layer identities.
That's fascinating.
Your brain was physically baking and refining those specific layer signatures well into your teenage years.
And speaking of baking those layers, there is a fascinating, almost detective story mystery hidden in the data regarding layer 4 specifically.
Oh, the layer 4 anomaly? Yes.
Yeah. The researchers found two completely different signatures for layer 4 neurons. There is P4, which is observed across all mammals, but then there is P19, which is a primate-specific layer 4 signature.
Which naturally brings us right back to the mouse question. If P19 is a primate-specific layer signature, what is the mouse doing with those genes? Or are they just sitting there completely dormant?
Right, and to figure this out, the researchers had to bring in spatial epigenetic data. They couldn't just look at the genes. They had to look at the chemical tags that physically bind to the DNA to turn genes on and off.
And the mechanism they found is staggering.
It really is. The mouse isn't just lacking the P19 pattern. The mouse biology is actively using repressive epigenetic marks to completely silence those specific genes in layer 4.
It's an active physical suppression.
Wait, so it physically padlocks the DNA?
Exactly. The chemical tags lock the DNA down so the cell's machinery cannot read it. Evolution has literally put the brakes on that complex transcriptomic program in the mouse.
That is unbelievable.
The genetic potential might be sitting right there in the DNA, but the epigenetic machinery is actively holding it back to maintain the simpler, smoother mouse cortical structure.
Okay, that's just incredible. Evolution differentiates species not just by adding genes, but by putting physical padlocks on complex programs.
It's elegant, in a way.
So just to recap this piece,
Right.
we have the ORGs driving brain expansion, true layer identities taking years to mature, and evolution using epigenetic brakes, like in the mouse layer 4.
Spot on.
Okay, so if we theoretically know all of this, if we know the exact assembly line, the repurposed evolutionary upgrades, and the epigenetic brakes, does that mean we hold the recipe book? Can we actually bake the cake ourselves in a lab?
That is the ultimate test of the NeMO Analytics Compendium. Researchers are currently growing cerebral organoids, basically, lab-grown mini-brains in petri dishes.
Right.
And to see how good these artificial brains actually are, the project utilized a tool called Project R to do something called transfer learning.
Right, transfer learning. Think of this like taking a highly refined, perfectly calibrated grading rubric developed in one classroom and applying it to grade tests in a completely different school.
That's a great way to look at it.
They took the in vivo patterns, the real-life natural blueprint we've been unpacking, and projected them directly onto the transcriptomic data from these in vitro lab-grown cerebral organoids. They literally graded the lab brains against the real thing.
And the report card is a profound mixed bag.
Let's start with the good news.
Okay, the good. The organoids get a solid passing grade for the early developmental strokes. They successfully navigate the early assembly line. They go from P5 to P4 to P7 to P2 in the correct order.
Okay, that's impressive.
They even show the early precursor patterns appearing well before the later complex outer radial glia patterns. They get the foundation right.
But the bad news, when that grading rubric looks at the highly specific 20-pattern adult layer identities, the ones we just learned take years or even decades to bake in a real human teenager, the organoids fail completely.
Completely.
Many of those layer-specific transcriptomic programs for mature neuronal identity are just entirely absent.
Which brings us to the real-world clinical stakes of this data. Right now, pharmaceutical researchers across the globe are using these lab-grown cerebral organoids to model complex neuropsychiatric diseases and test new drugs for things like schizophrenia.
Diseases that we know for a fact rely on fully baked, complex, intricately mature neuronal layer networks.
Exactly. This raises a really important question. If you are testing a novel schizophrenia drug on a lab-grown organoid, you absolutely need to know that your biological model hasn't actually developed its mature layer identities yet.
Right. The organoid is essentially stuck in a perpetual early embryonic state.
Precisely. If we don't acknowledge the biological limitations of these in vitro models, we could be throwing away highly effective drugs or wasting billions of dollars pursuing dead ends simply because the brain we tested them on hadn't finished baking.
So what does this all mean for us? When you step back and look at the sheer scale of the NeMO Analytics project, it's clear this isn't just a static table of contents for biology.
No, it's a dynamic living map.
Right. It shows us that building the most complex structure in the known universe isn't a series of binary switches. It's a continuous, overlapping, cross-species symphony. It relies on a delicate 4-day rapid groundwork phase, followed by a protracted, decades-long maturation process.
And, the most vital part of this entire compendium is that it is completely open source.
Really, anyone can access it.
Anyone. A geneticist in Tokyo, a bioinformatics student in London, they can upload their own single-cell data or their own gene lists and test their theories against the entirety of public neocortical data. They can project their own individual melodies to see if they play in the grand universal symphony of the mammalian brain.
Which leaves you, listening right now, with a final mind-bending thought to mull over. We've seen how evolution built the staggering, beautiful complexity of the human brain simply by repurposing old existing genetic programs like the mouse's support cell pattern, and by physically lifting the epigenetic brakes that were holding back complexity in simpler mammals.
Yes.
So if that is all it took to bridge the cognitive gap from a mouse to a human, what dormant programs might still be hiding in your own human DNA right now?
It's entirely possible that the blueprint for something vastly more complex than the current human neocortex is already written in our cells.
Just locked behind epigenetic padlocks, waiting for the right evolutionary pressure to unlock the next level of human cognitive ability.
Just waiting for evolution to remove the padlock.
Keep questioning, keep exploring, and we will catch you on the next deep dive.